All right, very good morning, everyone. Welcome back. Now we are continuing our presentations for the proteomic data analysis projects done by all of you, the students of this course. Today we have 5 presenters who are going to talk about their work, how they took the raw data from different mass spectrometry-based experiments and investigated different problems quite independently and try to come up with their own solutions about these issues, right? Let's first start with Ramya. And this topic is about acromegaly and Cushing's. These two are different type of pituitary tumors. And then here you are comparing within two diseases, what can be the possible way of identifying the potential protein and classifiers using ML approaches. So let's listen Ramya's project now.
Hello everyone, this is Ramya Dukirala, and today I am presenting my project on global proteomics analysis of acromegaly versus Cushing syndrome using machine learning. One of the previous projects presented did discuss about acromegaly. Just to recap, it is a disorder that occurs when there is too much growth hormone in adults. It is mostly a benign tumor in the pituitary gland, which is known as an adenoma. The symptoms include enlarged limbs, enlarged facial features, oily or coarse or thickened skin, abnormous sweating, strong body odor, small outgrowths of the skin such as skin tags, weakness and fatigue in the muscles, a husky voice because of enlarged vocal cords, severe snoring, impaired vision or frequent headaches, joint pains, and a barrel-sized chest. For illustration, there is a comparison between a normal hand and an acromegaly affected person's hand. Coming to Cushing syndrome, it is a condition that occurs when there is high cortisol for a long time. Cortisol is the stress hormone. Again, a couple of the projects that we discussed in previous day were about Cushing's. So I will not again bore you with the same symptoms, but as you can see, Cushing's also has a fatty hump between the shoulders and overall enlargement of the face and some stretch marks in the skin. It can also lead to osteoporosis or hypertension or type 2 diabetes. And again, there is loss of muscle mass and strength in this syndrome. So when somebody goes to a doctor, say, trying to address these symptoms, mostly it would be difficult to say at a glance that if a person is affected by acromegaly or Cushing's. Also, both of them being adenomas in the pituitary gland, it might be difficult even with some scans to say that exactly if it is acromegaly or Cushing's. But definitely, because both of them have different causes, the treatments should be different. So identifying suitable protein markers through machine learning might help us in the timely addressal of these fatal disorders. So for the data analysis workflow, I chose to first go through meta-analyst and then use the data in orange and then use the most regulated proteins from meta-analyst and orange and string and reactome to find some correlation or to find the right pathways. the correlation of pathways. The data set specifics and the data normalization are as shown below in meta-analysis. I was given 42 samples, 25 of which were of Cushing's patients and 17 of acromegaly patients. The number of features or the number of proteins that were chosen was around 4,250. There was no missing value in the data given to me, and there was a single outlier. While performing the normalization, I did not choose any scaling, and I chose to normalize by the median, and I chose to do log transformation. So this is the meta-analyst outlier detection. And with the confidence score plots, there was one single outlier. So that is what I mentioned in the previous slide. The VIP scores are as shown here. Here is a snapshot of my orange workflow. Built 3 models, one random forest, one SPM, and a neural network. The same confusion matrix was obtained for all of my models, which means that all my models were both accurate and precise. The string analysis of the top 15 ranked proteins using orange for Cushing syndrome yielded the following. The top 15 ranked proteins for Cushing syndrome from my orange analysis were as shown beside. And the top 15 ranked for acromegaly are as shown beside. So I could not find strong correlation between for the proteins in the acromegaly data, but for Cushing's, there was this single correlation that I could find using string. So these are the 2 proteins. One is ERKCA and the other is ARHGF1. And these are the details that I could find on string. So the reactome analysis for the protein significantly upregulated in Cushing syndrome are as shown here. For p-value less than 5%, 29 significant pathways that were identified, and out of these 29, only one of them, the neutrophil degranulation, had 3 entities. So these are the 3 entities. Neutrophils are critical inflammatory cells that can cause tissue damage in a range of diseases and disorders. Being bone marrow derived white blood cells, neutrophils are critical inflammatory cells that can cause tissue damage in a range of diseases and disorders. Being bone marrow derived white blood cells, they migrate from the bloodstream to sites of tissue inflammation in response to chemotactic signals and induce inflammation by undergoing receptor-mediated respiratory bursts and degranulation. Degranulation from neutrophils has been implicated as a major causative factor in pulmonary disorders, including severe asphyxic episodes of asthma. However, the mechanisms that control neutrophil degranulation are not well understood, so they could very well have something to do with cortisol-related pathways, making them prominent in Cushing syndrome. To conclude, while no significant insight was made with the acromegaly data, the Cushing's data promises hope in relation to neutrophil degranulation. The protein with the accession number E12111, which is called collagen alpha 3,6 chain protein, it is common in both the VIP plot of the better analyst and the top 15 rank of Cushing's using orange. So that could be monitored when one wants to track Cushing syndrome long term in a patient.
All right, Ramya, are you there? Can we now talk to you? Is Ramya there? Well, if a student is absent for the Q&A, then it will be difficult for the giving the marks for that part, but we have already assigned 3 marks just for the Q&A. But let me just give some broad comments in case Ramya is listening to me. So while it was the overall, correct workflow being used to look for comparing Cushing and acromegaly, major drawback I found that the analysis for the meta-analysis when you showed was not very clearly spelled out the details like what normalization you use, what kind of cutoffs were used, how you ensure the data quality, what the fold change p-values were used. None of those were actually showed, any analysis was shown. And same goes to the orange analysis, which type of models were selected, which models were actually used further to select that you are finding the best possible classifiers or top 15 candidates. Then of course other question which I had was essentially regarding here your major emphasis should be on comparing the two from the Cushing versus acromegaly. So if you identify, let's say, a set of 15 proteins from each one of these, the next thing should be that you need to do some blinded validation. Can you pick up the some more training data set and not pick up some validation data set to validate your findings based on the identified classifier and show that how sensitive and specific your assay or test could be based on these proteins. Also, the pathway analysis should have been done with more number of entities, not only the top 15, top 20. That's where in most of the pathways you see only one entity coming out, and that should be kept in mind by all the students. All right, so Ramya, if you missed out, I hope you will be able to see these comments in the recording. Let's move on to the next presenter now.
Hello everyone, this is Ramya Dukirala.
Sorry, so now we will be talking to Vinayak, essentially on the swab data of COVID-19 patients to look at the machine learning approaches, how that can be used to find the best classifier, understand the disease pathogenesis. I'll now play Vinayak's presentation.
Hi all, I'm Vinayak. I'm pursuing BTech in Computer Science at IIT Bombay. My project
voice is very low in recording itself.
Analysis of COVID positive versus COVID negative swab samples by machine learning approach to decipher the host response towards COVID pathogenesis.
Let me stop presenting once and see if there's any technical issue I am facing. Why your voice is coming so low? Vinayak, are you there? Do you see your voice is changed or not coming good here, or is that how the recording is?
Sir, it happened with the recording actually. The mic didn't take it properly, I think.
All right, well then I should not then play with it. Let me then share again.
So coronavirus disease 19 abbreviated.
Do you want to make a live presentation, because this is not going to make any impact, Vinayak?
Sir, if you put it at the max volume, it should help, I think.
Sorry?
If you put it at the max volume, I think it should help.
I have done the max volume, it's already max. Or let me also try out something here in the video.
No case was identified in Wuhan, China in December 2019. I'm pursuing BTech in Computer Science at IIT Bombay. My project is global proteomics analysis of COVID positive versus COVID negative swab samples by machine learning approach to decipher the host response towards COVID pathogens. Introduction of coronavirus disease 19, abbreviated as COVID-19, is a contagious disease caused by severe acute respiratory syndrome coronavirus 2, which again is abbreviated as SARS-CoV. The first known case was identified in Wuhan, China in December 2019, and the total count of cases currently is 242 million, and the fatality rate is 2%. So, the pathophysiology of COVID-19 is initially the inhaled SARS-CoV-2 binds to the ciliated secretory cells in the nasal epithelium, and after that, replication of the virus and local propagation happens with immune response being limited. And after that, there are two possible paths. In about 80% of the patients, the infection is contained and the viral clearance occurs in about 10 to 14 days. And in about 20% of the days, 20% of the people, the virus migrates through the conducting pathways into the lower respiratory tract, and after that, the pulmonary alveolar epithelial cells are infected. And after that too, there are two pathways which lead to the same thing, which is the diffuse alveolar damage with resulting acute respiratory distress syndrome, which is basically breathlessness. This is a symptom. So, talking about the preventive measures, maintaining the safe distance from others at least 1 meter, even if they don't appear to be sick, is a good thing to do. Wearing a mask in public, especially indoors or when physical distancing is not possible. Cleaning your hands often. Use soap and water or an alcohol-based hand rub. Get vaccinated when it's your turn. Follow local guidance about vaccination and staying home if you feel unwell. These simple measures can help to contain COVID to a good extent. Some other things to do are mentioned over here. So the project workflow will be, I'll be initially using the data provider to put it into the metabo analyst to analyze different properties of the data and normalize the data. After that, I'll be using the normalized data to give it to Orange and train different machine learning models to predict the COVID positive or negative depending on the swab sample. And after that, I'll be giving the top identified proteins to string, identify top pathways using reactor. So the data given to me had 36 samples, out of which 18 were COVID positive samples and 18 were COVID negative samples. 3,749 proteins were identified with 72% missing values were there. Missing value imputation was done by using feature-wise KNN, and normalization was done by using median, and log transformation was also done. So the figure here shows how the data was before normalization and how it was after normalization. As we can see, the data is distributed without bias in the after normalization. These are the PCR results in the metabo analyst. The first is the scores plot. So there are only two outliers without 95% confidence interval, and most of the points are falling in the required region. And these are the variances between different components. So the component one has a variance of 20.2%, second has 11.6%, the third as 4.2, the fourth is 5.6, and fifth is 4.7. These are the top 15 features via VIP scores, which is a measure of importance in the PLSDA. These are the top 15 proteins. And this is the Volcano Plot with P threshold kept at 0.05. Using this, we were able to see that 21 were significantly down-regulated, 38 were significantly up-regulated, and 263 were insignificant. This is the correlation heat map using Pearson score. So this is the correlation heat map between samples and so the samples are labeled from S1 to S36. So S1 to S18 are the negative samples and S36 to S19 are the positive samples. Like samples are grouped together. So S19 to S36 those are grouped together and these are grouped together. This is a heat map using Pearson score. Next, I use the data and transpose it to get it into a format which can be used by Orange to train the models. And in Orange, to get the top 5 attributes, I used information gain ratio and chose the top 5 proteins. At the first step, the rank. And after that, the ML models which I used were neural network, random forest, k-nearest neighbors, and support vector machine. For correctness about the predictions, I used the confusion matrix to determine the accuracy and false positives or negatives if any. So 60% of the data which came out from 22 samples was users training data and 14 were users data. And the next step is the parameters which I used with the machine learning models. So for neural network, I used 100 neurons in the hidden layer and the activation function as ReLU with Adam optimization solver and the max iteration set to 200. And the accuracy I obtained was 13 out of 14. 13 out of the 14 I was able to predict correctly, which came out to be 93%. And random forest, for the random forest, the number of trees I put 10 and the minimum subset split size was 5 and the accuracy I obtained was 14 out of 14. All the samples were predicted correctly. For the k nearest neighbors, the number of neighbors was set to 5 and the distance metric was set to Euclidean and the weight is set to uniform. The accuracy obtained was 11 out of 14, which is 78.6%. For this support vector machine 2, the cost was set to 10 and the regression loss to 0.1 and the kernel I used was RPF. The accuracy obtained was again 11 out of 14, which is 78.6%. So takeaways from the orange analysis was the best performing model was random forest, which gives an accuracy of 100%. Below is the confusion matrix for the same. So 8 of the samples which were negative were predicted negative correctly and 6 were predicted positive. The top 20 proteins from orange and metabolism. So I checked for some common proteins between the top 15 VIP scores in the PLSDA and top proteins in orange too, and arrived at this set of 20 proteins. The proteins were given to reactome to obtain the pathways. So these were the pathways, these are the pathways. And I gave it to string to analyze the correlations. So we can observe that there are three clusters. So going for the literature, we can see that we noticed TLR over here. So the TLR was present over here. So I was able to find a paper correlating both of them. The important points from the paper are the introduced hypothesis, the hypothesis introduced in the paper, sees TLR9 as a promising multifaceted intermittent target for prevention and treatment of COVID-19, and the readily availability of TLR9 modulating drug candidates that have reached clinical testing for other disorders could help a faster development for a vaccine for COVID-19, since the unmet need is very high currently for COVID-19. So our machine learning model has been able to perform, has been able to correlate something with the existing data. Thank you all.
All right, Vinayak, you are there?
Yes, sir.
Okay, can we talk to you?
Yes, sir. I'll turn on my camera.
Yes, can you turn on your video?
Yes, sir. I've turned it on. I think it's taking some time.
Okay, so your voice is very good, and you've done a good project as well. Now, please don't compromise on the quality. If you already knew that your video has low voice, you could have re-recorded it. Because after doing such a nice project and doing a lot of analysis from metabolism to orange to all the pathway analysis, everything which could have been made very impactful, and you identified some really nice targets. But it was not at all audible clearly. So we could make sense of the data based on what you are showing on the PPT, but as a presenter, what your explanations are were really unclear to me, and I'm sure to all the other students as well. Coming to your first analysis on the metabolic list, what was the missing value you obtained and how you imputed that?
I imputed the missing values using feature-wise KNN, and there were about 72% missing values in the data.
How much?
72%.
So that's really a huge number, right? So we have been also discussing that when you see this type of missing value, a huge percentage, do you also then try to look at the possible reasons for that? And now in this case, you had COVID positive and negative two type of populations, right?
Yes.
Did you look at them individually? Did you try to impute the values individually of each group?
No, sir. I didn't go through the data manually. Like I didn't do a sanity check or something like that.
Yeah, so that's I think something to be looked at, because it may also be that some changes are very reproducible in one population and missing another population, right? It could be 0-1 type of matrix.
Yes.
So
Yes.
yeah, so that's something was observed in your missing value part. When you were talking about various type of orange models in the neural network, you showed a term Adam solver. What do you mean with that, and how that works?
I think it's an optimization function to to calculate the weights of the neural network.
Do you have any room to change that or to optimize that? Or
Yes, sir. I think there are a couple more things, but like once I obtained high accuracy, I had not gone for the other ones. Like I tried to minimize the number of proteins to be used to get high accuracy. So I had used only 5 proteins to get that accuracy.
because you got the random forest making better working for you?
Yes, sir. With a small number of proteins, it was working well. So I kept them as default values.
So your final leads came as a TLR9 signaling as one of the key important findings, right, from your work. Yes. Now, some of the proteins identified from this signaling pathway, were they part of the best classifier which came from the orange? So that's something you should look into it, that because this pathway is coming interesting, whether the proteins belonging to this pathway, whether you have any idea for those pathways, and can you see, like, are they also the best classifiers, right?
Yes,
all right.
Sir.
You want to add anything or say anything on this point?
I looked at some proteins, like they had some correlation with COVID. Like one of them was the work calcium and zinc binding protein, and it played some role in inflammatory prostate immune response. So it had some role in COVID.
All right, thank you, Vinayak. So now let's move on to Shiva, who is going to talk about oral cancer and comparison with the controls to look at biological alterations using machine learning approaches.
Good morning, everyone. This is Shiva Taran of Computer Science Department, and my project is deep proteomics analysis of oral cancer tumor versus control, and decipher the biological alterations by machine learning. First of all, I'll talk about oral cancer. Oral cancer is also known as mouth cancer and is a major cancer, at least in our country, India. So this occurs usually in the linings of lips, mouth, and upper throat. And in mouth, it usually starts with a starts as a painless patch, white patch that thickens, and it becomes redder and redder with time, and it forms an ulcer and continues to grow. And on the lip, it commonly looks like a persistent cluster, a crusting ulcer that does not heal, and it grows very slowly. And other symptoms of mouth cancer are discomfort while swallowing, and lumps on neck, and swellings in mouth, and also a feeling of numbness in the mouth and lips. And here is a picture in which there are some symptoms of oral cancer, like ulcers which do not heal within 2 weeks, and all lumps or swellings in mouth, around head, mouth or head and neck area, etc. So, oral cancer. The major cause of oral cancer is because of alcohol and tobacco usage, and sometimes you can also get oral cancer because of HPV infection or chewing porn, and sun exposure on the lower lid. Diagnosis for oral cancer is made by taking biopsy sample of that cancer concerning area, followed by CT scans and MRI investigations. We can prevent oral cancer by avoiding tobacco products and limiting alcohol usage, and applying some protection creams on the lower lip, and also HPV vaccinations, and avoiding substances like pan masala, etc. And treatments for oral cancer can include a combination of surgery, radiotherapy, chemotherapy, and targeted therapy, depending on the stage, depending on the conditions. This is a project flow. Basically, I started with MetaboAnalyst. I did some analysis using MetaboAnalyst, and then during the analysis in MetaboAnalyst, I normalized the data. That data is used in Orange after some processing, and the outputs of Orange in MetaboAnalyst, the major proteins that we get as a result of MetaboAnalyst and Orange are given as input for string and reactome for further analysis. The data set I was given has 20 samples in columns, and the first 10 columns contain tumor samples, and the next 10 columns contain control sample. And a total of 19179 missing values were detected by MetaboAnalyst. So I did missing value imputation, and as part of missing value imputation, I used the following settings, where features with greater than 50% of missing values are removed, and the remaining missing values are estimated using KNN feature-wise imputations. Sorry, using KNN machine learning method, and After this processing of data and after the missing value imputation, the data is normalized using normalized by meaning it is log transformed. Initially there is a bias in the data, but after that, after the data normalization, you can see that you get a Gaussian like intensity. So this normalized data is passed for PC analysis, and you can see the first 5 principal components here, and here this is a 2D score plot, and if you can notice that there are no outliers in the 2D score plot. That means there is a clear difference between tumor and control. And this is the volcano plot that I obtained from MetaBoAnalyst. The following are the settings for the volcano plot. FC threshold is set to 2 and p-value threshold is set to 0.05. And it is observed that there are 165 significantly upregulated proteins and 137 significantly downregulated proteins, and 1033 unregulated proteins. See, unsignificant proteins, sorry. And this is the correlation heat map obtained. This is the entire complete heat map for the entire data set, and this is the top heat map for the top 25 proteins. Yeah. So in order to perform the machine learning analysis using machine learning, I used Orange workflow, and for this Orange app, the input is the normalized CSV file that was obtained at the end of analysis from MetaBoAnalyst. The entire data which is there in that file is transposed. That means the columns made into rows and the rows made into columns, and a target class, an extra column with 0 and 1 are added, where 0 represents that it is from the control sample, and 1 represent the other row is from tumor sample, and this data is given as input for Orange. In Orange, features are ranked using ANOVA. The top 3 features are chosen for training different machine learning models and test them for classification. I used 4 different machine learning models in total. The random forest, KNN, SVM, and a neural network were used. And to check the correctness of the models, I used confusion matrix. The following are the parameters for ML model. Okay, for KNN, the number of neighbors used are 5, and the metric uses Euclidean distance, and the weight of each metric is uniform. And for neural networks, number of neurons in the hidden layer of 100, activation function used is ReLU, solver used is random, and regulation factors is used 0.0001. SVM machine the cost is 1. The regression loss epsilon is 0.1. Kernel used is RBF function. And for random forest, number of trees are 10, number of attributes considered tree split is 5, limit of depth of each tree is 3. And the results obtained are these are the results obtained. Top 20 proteins obtained via analysis in Orange and MetaBoAnalyst t-tests. These are the top 20 proteins, and there is a perfect agreement between these two top 20 proteins which I got from MetaBoAnalyst and Orange individually. All these proteins are exactly matching, and they were also in the same order. And this is the confidence matrix I obtained from the machine learning models. I got the same confidence matrix, and you can see that there is a proper prediction. All the control model, all the control samples are predicted as control samples, and all the tumor samples are predicted as tumor samples. So this is the output which I got from the string analysis. You can see that there are the 3 clusters, and for the first cluster there are like 5 nodes, and in the remaining 2 clusters there are 2 nodes, and there are some other nodes which are completely unrelated with any other node. And this is the output which I got from the reactome analysis. And the significant identified pathways. These are the top 20 pathways that were identified as a part of reactome pathway analysis. I used these top 20 proteins as input for a string analysis and the reactome analysis, because we have like so many proteins, but if you do analysis for pathway and using all the proteins, it doesn't make any sense. So I used top 20 proteins because there was also an agreement between Orange and MetaBoAnalyst. So I used these top 20 proteins, and these are this is the output of string analysis, and this is the output of significant identified. So these are the significant identified pathways, top 20 ones. And after that, I did some literature review for some of the pathways, like a triglyceride metabolism basic triglyceride metabolism basic interaction and RHO GTP cycle, and there was like a good agreement between the results I got and the literature. These are some of the references that I used, and to conclude, proteomics analysis was able to classify oral cancer was controlled very effectively, and ML models that were able to produce models that were very accurate. And the pathways found during investigation are in agreement with the literature, as we and I've provided hyperlinked some of the papers here. If you click on this triglyceride metabolism, it will redirect you to the paper about triglyceride metabolism and its relation with oral cancer. Yeah, thank you.
All right, Shiv, are you there? Can we speak to you?
Yes.
Okay, so good. I think you did a project on the oral cancer, and that is one of the most prevalent type of tumor in Asian population in India. So looking at, when you analyze the data with MetaBoAnalyst, you had shown the heat maps, two heat maps on the left and right side.
Yes,
was there any difference in those two heat maps?
The right one is it is focusing on the top 25 proteins, and the left one is like in general, like the total one
with all the proteins.
Yeah.
But in addition to this detail, was there any other difference among the two plots which you had?
The first one is like little irregular, but in the second one you can see that the left part is little blue, and if you divide that into 4 quadrants, you can see that first and fourth, second and fourth are blue in color, and the first and third are red in color.
Correct. And then what could have been reason for it? Like what could have been the major change from the left and right?
That means these proteins are expressing differently in the tumor and the control.
Also, I think the major difference was there that in the left side, what you had the unsupervised cluster, those were all randomly classified with the oral control and the oral tumor. Whereas the right side was the supervised cluster that was based on the best features and also supervised. That's why you see that cohorts coming very differently, right? Blue and red. Okay. So coming to your final analysis from the orange and the pathways, do you see any correlation in terms of the best classifier which came? Are they mapping to some of the pathways like triglycerides and other which you have identified?
So I didn't get your question.
So let's say from your pathway analysis, you identified few pathways, including triglycerides, are coming most abundant, and that's probably very interesting in your thoughts, right, with your analysis.
Yes,
now, the best classifier which came are identified from the orange analysis. You had listed a list of 15-20 proteins from that, and on a list. What do you think? Are they really mapping to the first pathway? Then how many of those were there from the your triglyceride pathways?
So I was getting the same set of proteins from both MetaBoAnalyst and Orange, sir, and I don't exactly remember if they were mapping to that triglyceride catabolism and all.
Yeah, so when you look at any pathways, even you may not be from bio background, you may not want to look at the biology or clinical part of it. Still, at least whatever your best predictions are from the classifiers, are they really mapping to the meaningful pathway, right? So that's something to be looked at, because pathway analysis comes from the entities And then you should look at least is there any correlation, because once you highlight something, that should also map to the your best classifiers. At least some of them should be mapping to that. Right? Can you just name me 3 best classifier which came from your data, which was able to classify the oral versus the control?
And well class, all of them are working properly, sir. Are you talking about the proteins?
Yes, I'm talking about at least name 3 proteins.
I didn't search for the names. Matlab, I only remember the codes. That too.
Right. So I think even if, as I said, you may not be from this background, but still it's a good idea to then look at your top 3, top 5 proteins, look at some literature review and see, are they making sense? Are they really able to classify what you are actually, imagine the potential of this work, right? Oral cancer being one of the highly prevalent type of Asian tumor, and as you already mentioned, with the food habit and with the various smoking and other habits, it's really prevalent, right, in population. Is there a way to diagnose that early? Is that way to diagnose it more accurately? So, can some of your protein which you are identifying maybe potentially biomarker? You have no idea. And
Yes.
then it's important to look at literature and see, are they making any sense? And that's where your model also sometimes gets tuned based on what you find. If you're finding things which are not making very much correlation, they're not linked to the tumor, they're not linked to oral, then I think you might want to refine your strategies. You might want to look into other models as well, right? So these are the iterative process which has to go through your findings followed by literature, and sometimes literature helps you to fine tune your models and work further on that. Okay, all right. Well, good effort overall. So let's move on to the next presenter.
Thank you, sir.
All right, so next presenter is Karishma Ghosh. She is going to talk about colon adenocarcinoma, one of the highly prevalent type of tumor in the Northeast population. And I think Japanese population has the largest incidence of it. And after that, in the Assam, Aizawl, Mizoram, these places have the very high prevalence in India. So from this project, let's see what Karishma has identified, the best possible classifier and understanding the biological alterations.
A very good morning. Today, I, Karishma Ghosh, a student of MTech Biomedical Engineering second year, will be doing project presentation for the course BB607 Principles and Techniques. Topic for my project is global proteomic analysis of CAC versus CAC control sample by machine learning approach to decipher the biological alteration. The following presentation contains literature review, metabolome analysis, data processing and analysis, development of machine learning model using orange, pathway analysis using reactor, and the conclusion. Multiplex associated cancer or CAC. The development of colorectal cancer in patients with inflammatory bowel diseases is known as colorectal associated colorectal cancer or CAC. Colorectal cancer is the third most common malignancy and one of the major causes of cancer-related fatality worldwide. IBD or inflammatory bowel diseases includes ulcerative colitis or UC and Crohn's diseases CD. Chances of CAC development in IBD patients suffering from ulcerative colitis is 18 to 20% after 30 years. However, it is 8% for patients suffering from Crohn's disease. CAC is a classical inflammation-driven cancer. IBD or inflammatory bowel diseases always proceed sometime by taking tumor initiation in CAC patients. However, the same case is not true for CRC or colorectal cancer patients. CAC risk is directly correlated with the severity and longevity of the active IBD disease in a patient. An important hallmark of inflammation in cancer is actually the ability to cause mutation. Inflammatory cells such as activated macrophages and neutrophils are potent sources of reactive oxygen species and reactive nitrogen intermediates, which are highly reactive and mutagenic entities and causes DNA mutation and damage in a patient. The research lab. Genomic analysis of CAC has not been thoroughly investigated. Therefore, our underlying carcinogenic mechanism understanding remains unclear. Our understanding of general mechanism which connects inflammation and cancer in a CET patient is still unclear. The following project workflow has been processed. The dataset which was received was first uploaded in MetaboAnalysis, where single-value imputation has taken place. The samples were normalized and data was transformed. Then, various data analysis process was followed. The Multiplex IDs obtained from MetaboAnalysis was then fed into Lecture, where pathway analysis was done. The normalized data that we received from MetaboAnalysis was then scaled and prepared for uploading in Orange using MS Excel. The normalized data, which was also scaled using MS Excel, was then uploaded in Orange for development of machine learning models. The data processing was done using MetaboAnalysis. Here, the data processing information has been presented. We observed that two groups were detected in the samples. There were a total of 44 samples by 5,812 peaks data matrix that was uploaded. The samples were not paired. Non-numerical values were identified and were replaced by NA. A total of 60,222 missing values were detected, which was actually 23.5% of the total data that was uploaded. Next, missing value computation has taken place. So, any feature that had more than 30% of this value percent was replaced, and estimation missing value using CNN feature-wise was implemented. Next, the samples were normalized using median and data was transformed using log transformation with base 10. Data scaling in MetaboAnalysis was done. Here is what we observe the Gaussian distribution after the data has been normalized, and we see that we obtain the desired Gaussian distribution. And normalization basically helps us in preventing the run-to-run variation in a sample. Next, principal component analysis is done. Here we can see the principal component analysis, and we observe in the 2D code that 2D plot that there was an outlier, which is CRC9N, which was one of the normal samples, and the outlier was removed. Next, fold chain analysis. Here the fold chain threshold was kept at 2, and significantly downregulated protein observed were 2,265, significantly upregulated protein that were observed were 391, and unsignificant protein observed were 3,319. Next, t-test analysis was done. Here the p-value threshold was kept at 0.05. Significant protein observed was 1,301, and unsignificant protein observed was 2,674. Next, Volcano Plot was used, where both fold chain analysis and p-value threshold were implemented, and the significantly downregulated protein observed here was 254. Significantly upregulated protein observed was 398, and unsignificant protein observed was 3,423. Next, we use PLFPA for identifying the important features. We identify the important features using VIP scores. So the various VIP, Unicode IDs which are important, which can be taken as important features for further analysis, have been shown here. Next, I have actually taken the Unicode IDs and identified the protein and gene associated with it. The first one is calcium activated chloride channel regulator 1. Second is prochloride, zymogen membrane protein 16, etc. Next, machine learning model development was done using Orange 3.30.1. Here, the file that we has normalized using MetaboAnalysis, the output of the MetaboAnalysis file was first opened in Excel, and the data was scaled. The normal was scaled at 0, and tumor samples were scaled at 1, and then the data was uploaded in Orange. The features that we have identified using VIP scope MetaboAnalysis were selected, and the following models were run. Logistic regression, SVM, random forest, and tree were the models that were used here. And the output of all the 4 models were also stacked and run again in test and scope to see the to see the analysis of the stack data, and evaluation of the result was done using confusion matrix and ROC analysis. Here we can see the confusion matrix for SVM, stack, random forest, logistic regression, and tree. So we see for SVM, stack, and random forest, out of 44, 42 samples have been correctly classified, and only 2 of them has been wrongly classified. Whereas for tree, we see that 4 samples have been wrongly classified, but only 40 samples have been correctly classified. We take the analysis further and we see the test scores, and we identify the various parameters. Here we see that based on various parameters such as AUC, CK, F1, precision, and recall, that SVM is the most suited molecule. However, stack also comes very close to the scores of SVM. Next, we check the ROC plot, and we can see the area under different curves. We specifically check the ROC plot for SVM versus stack, and we see that the brown identifies the area under SVM, whereas green signifies the area under stack. And we see that more area is covered under SVM, and as we know, greater the area covered under the classifier, better is the classifier. So we deem SVM as the better classifier. Next, the uniform identities obtained in metaboanalysis was fed into Reactor for pathway analysis. And out of the top 25 significant pathways, the top 10 significant pathway has been mentioned here. Some of the top significant pathways are formation of tertiary complex and subsequently the 43X complex, ribosomal standing and start codon recognition, formation of a pool of 340S subunits, GDP hydrolysis and joining of the 60S ribosomal subunit, L30A mediated translation silencing, etc. The most significant pathway identified was formation of ternary complex and subsequently the 43X complex. Here, the binding of methionine, the RME initiated to the complex of results in the formation of the tertiary complex. The tertiary complex then binds to 48 subunits and other proteins to form 43S complex. Conclusion: colorectal cancer that develops in patients with IBD is known as colitis-associated colorectal cancer or CAC. Genomic analysis of CAC has not been thoroughly investigated, therefore the underlying principle governing carcinogenic mechanism remain unclear. Also, the mechanism that connects inflammation with cancer initiation in this particular cases is also unclear. The dataset that we uploaded in metaboanalysis showed, after processing, showed that there was 60,222 missing values, that is 23.5% of the total data. The data set that was uploaded contained 44 samples and 44 samples by 5,812 fixed data matrix was obtained. After volcano plot analysis, we observed 254 significantly downregulated and 398 upregulated proteins were identified. SVM was identified as the most suitable model based on AUC, precision, F1, recall parameter, and ROC analysis, and also confusion matrix. Based formation of tertiary complex and subsequently 43S complex was identified as the most significant pathway using Reactor. Thank you.
All right. Thank you, Karishma. So, are you there?
Yes.
Okay. All right. So, about this problem of colon adenocarcinoma, looking at your data, what you identified as the best classifier from the SVM analysis, that was probably most robust one what you did, right?
Yes.
Can you think about, are there some targets which are also reported in literature as a part of colon adenocarcinoma or colon cancer?
So some kinases enzyme has been reported, and P53 mutations have been reported in literature. Actually, the proteins that are identified were not actually correlating with the proteins and enzymes that have been mentioned in literature. So I could not exactly correlate them with the literature.
But did you make any effort of looking at your, let's say, top leads identified from SVM analysis, and then look into literature that what is the known, are they linked to any cancer or in some way with the oral cancer?
No, sir, I did not do that.
That's okay. I think, different people have different background here, and that's what we gave more computational projects as well. So at least looking at the meta-analysis data, which I think all the steps what you defined were quite accurate for both meta-analysis and Orange. And in fact, Orange, the 5 confusion matrix which you showed was quite clear as well. In terms of the correlation from meta-analysis versus the Orange, do you see there is some lead which is coming common as well from the both analysis?
Sorry, sir, I did not get the question.
Let's say meta-analysis gave you some top 20 or 25 as a VIP score protein, right? And the Orange also gave you a very important 15, 20 proteins as a classifier. So do you see some correlation coming, like some are common as well coming from both, and are they really important or influential proteins in the colon cancer?
So actually, I took the proteins at top 10 proteins that were identified in meta-analysis, and actually I selected those proteins for analysis, for the model analysis in Orange. So I did not actually use rank in Orange to identify further the top proteins using Orange.
But do you see, are there some common which is present in both the analysis, which is emerging from both?
No, sir. I did not check, co-check if there were some common proteins.
Right, because it always gives more confidence if you have the proteins coming from different type of analysis, and they show higher ranking. Because all of these are still computational prediction. Finally, think about the biologists and clinicians. They have to do many tests to validate these proteins. More number of protein you will give them, more tests they have to do. So therefore, smaller the number of proteins and more confident protein, that will be better for clinical testing as well. In terms of the pathway analysis part, you talked mainly about ternary complex as one of the key pathway emerged from your analysis, and look like you have done pathway analysis accurately using the entire data set of the significant proteins, right?
Yes.
That's where all the 53, 54 proteins were mapping to the reactome. Now, again, did you pay attention to some of the protein involved in ternary complex, and whether they were also found in your top proteins from the SVM analysis?
So, sir, I read the literature related to the formation of 43X complex and the ternary complex, and I could not identify any proteins with the top 10 proteins that I've identified in the literature. So, therefore, it was not mentioned in the presentation.
So your proteins which you came identified from SVM, where are they going? Which pathway they're going? That's something one need to also look at, because that's after all your computational analysis, you have narrowed down those proteins, right?
Yes.
So the next curiosity will be, most of my influential proteins are going to which pathways? So rather than looking at the entire significant list, which is fine, you are looking at the most abundant changes, but still you'd like to see where your highest classifiers are going to which pathways, and are there substantial number. For example, if 5 protein from SVM is going to one of the pathways linked to ribosomal scanning or some other type of ribosomal machinery, that might be more important than ternary complex for that matter, right? Because then you'll have high confidence in that pathway and data.
Yes.
Okay. All right. So good project, good analysis done. Let's now move on to the next student. Okay, so the last presentation is from Rohit Janajanam. Rohit, are you there? I have not received your video. It's comparison of acromegaly versus non-functional pituitary adenomas. Is Rohit there in the audience today? I have not received your video, Rohit. So if in case I do not hear back from you, I will continue right now. The short time left for us to give all of you some idea for an initiative of the Human Protein Atlas. And that's something which we could not cover, but that was a very important initiative. I'm going to show you a brief talk about HPA initiative and how that is influencing the biology and clinical findings directly from the director of this project, Dr. Matthias Ullen. And please listen this presentation for the short duration what we have.
Good morning, ladies and gentlemen. We are extremely delighted to welcome Professor Matthias Ullen. Professor Ullen received his PhD at Royal Institute of Technology, Stockholm, Sweden in 1984. After a postdoc period at EMBL Heidelberg, Germany, he became professor in microbiology at KTH in 1988. His research is focused on protein science, antibody engineering, precision medicine, and ranges from basic research in human and microbiology to more applied research including clinical applications in cancer, infectious diseases, cardiovascular diseases, autoimmune diseases, neurobiology. His research has resulted in more than 650 publications. His group was first to describe a number of innovations in science, including engineered protein A and protein G for purification of antibodies, affinity tag for purification of recombinant fusion protein, solid phase method based on magnetic beads for DNA handling using the Biotin Struct Evidence System, biosequencing leading to first next generation DNA sequencing instrument 454 Roche, Affibodies, protein binders aimed for therapeutic application. Since 2003, he has led an international effort to systematically map the human proteome and transcriptome to create a human protein atlas using antibodies and various omics technologies. This effort has so far resulted in 5 major launches, all published in the journal Science. The Tissue Atlas, showing the distribution of proteins across human tissues and organs. The Cell Atlas, showing the subcellular location of human proteins in single cells. The Pathology Atlas, showing how cancer patients' survival is tied to RNA and protein level. The Blood Atlas, showing the expression of proteins in human immune cells and also mapping the proteins in human blood. The Brain Atlas, showing the proteins expressed in different parts of human, pig, and mouse brains. Join me in welcoming our plenary speaker for the day, Professor Matthias Uhlin.
We can hear him.
Yeah, we can hear you. We can hear you. All of us can hear you. I have shared my screen on Teams. Yeah, you're good.
So I started by just showing that 20 years ago, the announcement of the human genome gave us a blueprint for human biology and diseases. But obviously, in order to study the future of biology, we have to study the proteins. And that's why you are on this workshop. So what we asked us 20 years ago, what are the building blocks of cells, tissues, and organs? And really, what we want to do in the end is, can we model human biology from its protein building blocks? And this is what we are trying to do. So I am the founding director of an institute in Stockholm called Science for Life Laboratory, which is a national infrastructure for life science. And this was started in 2010 to actually do big data analysis and technology-based infrastructure. We are now about 1,200 researchers, and last year we ran about 2,000 projects. Before I continue, I just want to say that I'm co-founder of several startups, and I will mention some of them in this talk, especially Antibodypedia, but I'm also doing collaborations with AstraZeneca. So what I thought I will do is to give you a little bit about the 20 years with the Protein Atlas, the new version 20, talk about precision medicine, and then talk about the movies that we made, which are journeys into the human body. So the Human Protein Atlas, the vision is to map all the human proteins using technologies to create an open access atlas of the human proteome for researchers in academia and industry. This is very much a European effort from Sweden, but we have very important collaborations with people in South Korea, BGI, but also the most important collaborator has been in India, in Mumbai, where and I will come back to this. So in Science magazine, some months ago, we published a digital booklet that takes you through the 20 years of HPA and the milestones. This is a 140-page document, so I'm not going to go into it in detail, just show you some of the highlights in this. One of them, it started back in 2000 with a chromosome 21 pilot, we decided to make antibodies to all proteins encoded from chromosome 21, and then to analyze protein profiles in tissues. 5 years later, we launched the first protein atlas with 7,000 antibodies, and we were very proud to have 400,000 immunohistochemistry images in that launch. But then we started a collaboration with Sanjay Navani and colleagues in Mumbai, and we set up then a site in Mumbai, in central Mumbai, for high quality pathology annotations. And this has been a fantastic collaboration between the HPA and the Indian site. And the site in India has annotated more than 10 million pathology images. And I think that's the largest number of annotations in the world. In 5 years later, 2015, we published the first major paper in Science, the Tissue Atlas, and it is actually one of the most cited research publications from Europe in the last 6 years now. So we're very proud of that. But since then, we have published quite a few additions to the Protein Atlas, a subcellular atlas in 2017, a cancer atlas in 2017, a blood atlas about 2 years ago, well, actually 1 year ago, and then a brain atlas in April last year, and a metabolic atlas. And in total, more than 550 publications from our group. So what have we achieved then? We have generated ourselves about 55,000 antibodies. 21,000 of those has passed the stringent requirements, and we have created more than 10 million immunohistochemistry. And we've done a lot of validation of antibodies, more than 70,000 experiments. So we're very proud to have generated more than 10 million images using these antibodies. And it makes us, I think, the largest effort in the world to generate tissue images. And each one of those images has been annotated by a pathologist in India. So we're very grateful to that. So the data we have in the Protein Atlas now is open access. We have more than 300,000 visits every month. And it is one of the 3 largest biological databases now in terms of visitors in the world, actually. And we're very, very happy for that. So what have we learned? Well, we have learned that about 10% of the proteins are what we call housekeeping, and thus needed in all dividing cells. About 15% of the proteins are specific and present in only one or very few tissues. And about 13% of the proteins are secreted. We call it the human secretome. And these are, of course, very interesting for all of us that are looking at blood proteins. We also made a lot of movies, and I thought I would show you some of these, and I hope that the sound actually comes through. And the first one I want to show you is a movie. And we have made this using a new technology called iDisco, using antibodies. And so these are real images inside the human body. And we're going to start with a beautiful, stunning movie of the inner ear of humans.
This is the inner ear. It is one of the smallest, yet one of the most important organs in our bodies. The functions of the inner ear are multiple. It consists of the cochlea, responsible for our hearing, and the vestibular system, responsible for our balance. Damage to the inner ear can not only lead to a loss of hearing, but also imbalance, tinnitus, and vision impairment. With these 3D images, it's possible to really appreciate the beauty and complexity of the inner ear.
So I just wondered, could you hear the sound of the video?
Yes.
Okay.
Yes.
Well, yeah. Very good. Thank you very much. So a few months ago in November 19, we released the version 20 of the video. We had two major new parts of it, a single cell type atlas and a prefrontal cortex atlas. And I want to quickly go through that. So we have used then the RNA analysis and the new single cell analysis from human tissues to try to integrate that with antibody-based profiling. And we have been looking at 192 single cell types and actually analyze what proteins and RNA are in these cell types. And just to show you one example, this is the PSA. You can then get these kind of clusters. We call it, this is the prostate, and every point here is a single cell, and then it says how much in these cells is the PSA expressed, and you can see there are two clusters and they happen to be glandular cells. And then you can actually go in and look at this with the antibody in the protein atlas. And all of this data is available in the Human Protein Atlas. You can also go in and look for the classifications. So here, for example, we are showing all the tissue-specific or cell type-specific proteins. And we can, for example, see that there are 231 proteins which are specific for early spermatids in the germ cells. And there are 800 proteins which are shared between late and early spermatids. And then you can click on this bubble and then you can see what proteins it is. And I hope that you all can go and do this kind of analysis. It's also interesting to look at proteins which share expression. And for example, we now have the proteins which are shared between ciliated cells, lung, and testis. And this obviously, when you look then in the protein atlas, you can see that this is ciliated cells in these two tissues. So we're trying then to integrate the single cell analysis with antibody-based profiling, and we also do quite a lot of proteomics, mass spectrometry. I will not talk about that today since the whole workshop is on that. We have then moved on and are now doing a lot of multiplex tissue profiling. So we're combining single cell data with the multiplex in immunohistochemistry. And just to show you the beauty of this, I show you a kidney stained with 6 antibodies. And you can clearly see then the glomeruli here in sort of red. You can also see here both the proximal tubuli, the distal tubuli, and the collecting ducts in green. And you can see very, very nicely where your protein is localized. We also are working a lot now with detail of the human brain. And what we've started with is the prefrontal cortex, which is a very important part of the human brain. And I'm not going to go into the details here. This is people at the Karolinska Institute. But we are now creating a very detailed molecular map of all the different regions of the prefrontal cortex, but I don't have time to go into that. But what we're doing is do microdissection of brain regions using a punch needle approach. What we're also doing now is to look and do precision medicine. And the idea here obviously is to get the right treatment to the right patients, to do more targeted treatments and better diagnostic methods. The main way of doing that is we're using targeted proteomics, we're using mass spectrometry, but we're also doing it using antibodies. And we are using now a very new technology called Olink Explorer that allows us with very low sample volumes, 5 microliters of blood, to have a look at about 1,500 targets with very high sensitivity. We can go down to cytokine levels. And this we can do in one multiplex assay. So this is quite a fantastic technology. And we hope to have a paper coming out very soon in Nature Communications. So with that technology, then we have then looked at different patient cohorts, and this just shows you the Swedish copy Scilab Lab Wellness Profiling Program, where 100 individuals has been followed for 2 years. We're taking samples every 3 months, and then we analyze them with whole genome sequencing, Olink Explorer, target proteomics using mass spectrometry, immune cell profiling, metabolomics, again using mass spectrometry, gut microbiota, and so on. So these 100 individuals, I think, is the best studied individuals in the world, actually. So what you can then do is that you can look at the protein levels. In the middle here, you see the famous leptin gene or protein in the human plasma. And here you can see in gray, you can see the individual, how they are, the concentration in the blood of individuals. The average for females is here in blue, and the male here. And you can see very clearly then that females have higher levels, but also very interesting is that the individual levels vary quite a lot, both in the female and male, but they are stable during these 2 years that we have sort of studied it. So there is large individual variations, but the actual individual variations are stable during 2 years. And this is, of course, very, very interesting for all of us. You can also do this kind of dynamic UMA plots. Each dot here is an individual, female in red and male in green. And this is then how the whole profile of the proteome is changing during 2 years. And you can see for most of the individuals, they have an individual profile, as I said, but it's stable. They are sort of stable during 2 years. But then there are actually some individuals that are changing a little bit during these 2 years. You can also see on this other omics that they are much more variable. So the proteome is quite stable. We've also been doing whole genome sequencing of this, so we can then compare the genetic variants of these of these individuals with the plasma profiling levels. And it is then interesting to see that some proteins are completely determined by genetics. So you have, for example, this famous receptor PDGF. You can see then there is a variant in the gene where you have the CC variants have lower levels than the TT variants, and the heterozygote has about the same. And you can also see that very clearly here. So there are proteins then that you're born with a genetic variant, and that determines your adult levels of that protein in the blood, which is, of course, very interesting.
All right, so you just heard Dr. Mathisulan and very impressive Human Protein Atlas project glimpses. Complete lecture is available in our YouTube. You can watch that. You should definitely explore the HPA website and look at how well you can look at some of the identified protein which all of you have identified from your projects and see where they are localized. Are there also correlation of these protein, what you do from mass spec at the different technology level, looking at the immunohistochemistry, looking at the other type of tissue arrays. And that's where you get more data confidence by comparing your data to other data sets. That's all for the day, but this last lecture, the glimpse of it will be part of the writing assignment as well, other than the 4 presenters who were presented. This will be treated as the fifth presentation, and it will be part of the writing assignment. We'll close here. We will meet again on next Wednesday and finish the presentation. Then I'll move on to new technologies like protein microarrays and some of the label-free biosensors. Thank you very much, everyone.